So today we're going to be hearing from Dr. Josh Bryan. Josh is a community ecologist with an interest in biological invasions and parasites. And he's going to be talking to us about parasites in biodiversity conservation and whether they are a friend or a foe. He's originally from Wellington, New Zealand, where he completed a PhD in aquatic ecology. And he's come to the UK to study at King's College London, working on the Alien Impact Project to predict the impact of alien plant invasions, and really looking forward to hearing from him. So I'm just going to hand over to you now, Josh, to start your lecture.
Lovely, thank you very much. Well, hello everybody. Thank you for coming, or virtually at least, to my talk today on parasites in biodiversity conservation. Are they friends or are they foes? Hopefully it's an interesting topic for you all, and I've tried to get some interesting natural history in there as well. Thank you also to the Linnean Society of London for inviting me to give this talk. I'm currently a postdoc, as Anna said, based at King's College London, but most of what I'll talk about today came out of my PhD, which was funded by New Zealand funder, the Wolfisher Trust, at the University of Cambridge. So I'm not sure how aware all of you are about the commonness of parasites, but they really are the unseen majority in the world's biodiversity. More than 75% of food web links are thought to involve parasites, and they also have this massive amount of biomass. So for example, in Oregon streams, the biomass of parasites, just of trematode parasites, was found to be greater than that of all aquatic insects combined. So they're a massively important and common player in biodiversity conservation potentially. So the question is, how do they interact with conservation aims? So I'm going to take you through some examples of how parasites interact with conservation in both positive and negative ways, and I'll largely focus on freshwater mussels as a system to explore these ideas, as this was the focus of my PhD, but I'll also include some other examples where appropriate. So freshwater mussels are hugely important. You can see me there in my slightly younger days, proudly holding some freshwater mussels from the first lot of sampling I did for my PhD. And these mussels are incredibly important in the environment. So these are two photos that you can see there of the River Cam taken on the same day, but the photo on the left is a bit of the River Cam that has freshwater mussels, and the photo on the right is a part of the river, the little tributary, that doesn't have freshwater mussels. And you can see the massive difference, again, taken on the same day. The one on the left, nice and clean and clear water, the one on the right, choked with algae. And that's because freshwater mussels provide this incredibly important filtration role. So they're incredibly prodigious filter feeders, and in doing so, they basically clean the water and deposit any sediment then on the bank or on the riverbed, rather than it floating around in the water. So my favorite fact about freshwater mussels is that the entire volume of the River Cam is filtered through freshwater mussels every 2 weeks. And you can see the importance of their role in those photos. They also play a whole lot of other important roles. They form these really dense mussel beds, so they actually stop erosion, stop the riverbank collapsing into the water, and they provide habitat for small fish, aquatic insects, other aquatic life, things like that. Freshwater mussels are also highly threatened, hence the relevance of thinking about their parasites. They're actually the most endangered non-marine group of animals on Earth. For example, up to 60% of North American mussels are threatened with extinction. And they're faced with a whole range of threats, like water abstraction, dam construction, pollution, anoxia through more nutrients coming to the water, increased temperature of their rivers, all these things. And one of the questions that I had was, could parasites also be a threat to these freshwater mussels? So parasites generally are thought to be a major cause, potential cause of extinctions, both on a global level and a population level. So they're potentially implicated in some global extinctions. For example, it's thought that some amphibians, some undescribed amphibians, might have already gone extinct without us knowing, thanks to the chytrid fungus, which is this massive fungus which is spreading through amphibian populations worldwide, with the massive influence of climate change, of course. But parasites also cause population level extinctions. So certain populations can go extinct if they suddenly get a parasite. For example, there's a microsporidian parasite currently spreading through the Mediterranean, which is causing some populations of fan mussels in the Mediterranean to just completely die with 100% mortality. And so given the relevance of freshwater mussels for freshwater ecosystems, one of my key questions was, could this happen for freshwater mussels? And one of the key focuses in terms of their conservation is obviously reproduction. If we have a nice, healthy, reproducing freshwater mussel population, they're more likely to be able to survive and handle parasites. But parasites might actually affect the reproduction of these mussels. So that's what I'll talk about now. And first, to just give you a very brief introduction to the reproduction of freshwater mussels, because it becomes relevant in a second and also later in the talk. These freshwater mussels are actually amazing. They do a really interesting thing with their babies. So the female mussels, they have eggs in their gonad, and they will inhale sperm through their inhalant siphon. So the males will release sperm into the environment. The females will then suck up that sperm, fertilize the eggs in their gonad. And what the females do is they move their eggs to their gills, and they have these special water tubes called marsupia in the gills of the mussel, and they put their little eggs there, and those eggs develop into baby mussels called glochidia, like you can see in the picture there. And this is called gravidity. So when the mussels have their gills filled with these little baby mussels, they're called gravid. And then eventually, after the glochidia, the little baby mussels have developed in the mother for a period of time, they'll then expel them into the environment. So this is a really unique, really interesting reproductive system. And trematodes might affect this. So trematodes also have this incredible life cycle. So these are digenean trematodes that we're focusing on here. So they have this 3-host life cycle. So the adult trematode, if you look at the little diagram on the right, first of all, the adult trematode lives in large freshwater fish. Those adults will sexually reproduce in the fish. They will release eggs into the water, which then become this little swimming stage called myricidia. Those myricidia then infect the mussel, where they develop into these sporocysts. So that top photo on the left, those are sporocysts inside the gonad of a freshwater mussel. So these sporocysts reproduce asexually. So they basically spread throughout the entire gonad of the freshwater mussel. When those sporocysts are mature, they actually swell up, and inside them, they produce cercariae, which you can see there on the bottom photo on the left. Now, I think rather unkindly, I call these the dick and balls of the parasite world, because that's exactly what they look like. I'm not sure if that's ever been said before in a Linnean Society lecture, but there you go. So those are the next infective stage of the trematode. So those cercariae of the trematode will then release themselves from the mussel, then infect a small fish, then gets eaten by a big fish, and the cycle continues. So in mussels, this is a lifelong infection. So once they're infected by these sporocysts and producing their cercariae, the mussels can't clear that infection. They will be infected for life. And so for essentially, for the rest of the mussel's life, it becomes a trematode factory. And this can still go on for quite a long time. The trematode, under normal circumstances, doesn't affect the survival rate of the mussels. So the mussels, some of these mussels can live up to 20 years. So if they're infected, the first couple of years of their life, they might have nearly 2 decades of just producing these trematodes. And what this does, because these trematodes fill the mussel gonad, it basically castrates the mussel. So if we look at, these are 2 biggest sites from my PhD. So BC is Brandon Creek, and OW on the graph there is Old West. So these are 2 tributaries of the Great Ouse River, which runs through Cambridgeshire. And what I did is I looked at mussels in these rivers that were and weren't infected with trematodes, and I saw what proportion of the mussels were gravid. So these female mussels with the little baby glochidia in their gills. So you can see the mussels that didn't have trematodes, those grey bars there, in both sites, about 50% of the mussels on average were gravid. But the mussels that did have trematodes, you can see there was no mussels at all that were gravid. So these trematodes basically completely castrate the mussel. There's absolutely no reproduction happening from the mussels there, which obviously constitutes a rather major threat to their conservation potentially. There are other parasites as well, which might affect the reproduction. So another one is mites. So that's a unioniclid mite. That photo on the left looks rather terrifying, but again, that scale bar is about 100 micrometers, so they're really not that big. But these mites live in the gills, which is where those baby mussels also hang out, and these mites eat gill tissue, so they might also affect the reproduction of mussels. And indeed, that's what I found. So here I was looking at the percentage of glochidia that were viable. So mites didn't affect the production of the Glochidia, but they did affect the survival rates of the Glochidia when they're sitting inside the gills of the mother mussel. The way you can test this is just with salt. So you can very carefully extract these Glochidia alive from the gill of the mussel. You put them in a little bit of water and add salt. And if the Glochidia are viable, if they're nice and alive, you'll see this really nice snapping motion because they don't want to be in salt water, so they'll snap shut. And that's how you tell if they're viable or not. If they're not viable, they'll just sit there and basically be very unhappy. And what you can see is that the presence of mites actually affected the percentage of Glochidia that were viable. So overall, in mussels without mites, about 75% of the Glochidia that I found were viable. That's pretty good. But in mussels with mites, you can see it reduced viability to about 50%. Interestingly, this was only observed in one site. So in the Old West River, there on the right, mites had no influence on viability, but there on the left in Brandon Creek, they did have a negative effect on viability. So this suggests that the effects of parasitism might be context dependent, and I'll get to that again in a second. In addition to directly harming the reproduction of mussels and of organisms generally, parasites could also potentially amplify other stresses and could harm conservation efforts through these indirect effects too. So this was an experiment that was done in the mid-2000s, looking at anoxia, so basically oxygen deprivation. This is a major threat that face freshwater mussels. And so this might interact with parasitism. So you can see that anoxia, as they increased anoxia, the survival rate of mussels without trematodes actually didn't change. So mussels, unparasitized by themselves, they can actually deal with anoxia all right. But the survival rate of mussels with trematodes significantly decreased as they increased anoxia. So again, this is this interaction of multiple stresses. By themselves, trematodes don't kill mussels. By itself, anoxia doesn't kill mussels. But you get both together, and suddenly you get this increased mortality in freshwater mussels. And given the prevalence of global change drivers and other environmental threats happening to freshwater mussels and to all organisms, this is really concerning that parasites might interact with these other stresses and amplify these other threats. So the question is, can we predict these negative impacts? Do we know, will they be ubiquitous? Will they be in certain populations? And the answer is really no. So in this particular study that I did at Brandon Creek and Old West, you can see Old West had nearly 3 times as many trematodes, over 5 times as many mites. And then also there on the right, we've got zebra mussels. You might have heard of zebra mussels, these incredibly invasive mussels. These are also ectoparasitic on freshwater mussels. So they will live on the outside of freshwater mussels, attach themselves to the shell of the native freshwater mussel and basically steal their food. So they'll all the food that's coming into the native freshwater mussel, zebra mussels will intercept those and get it for themselves first. So you can see mites and trematodes were much more common at Old West, but the zebra mussels much more common at Brandon Creek. I'm sure you're probably wondering now, why is this? The answer is I have no idea. Can't answer it. There was no correlation with any measured environmental variables. We really don't know why these parasites are distributed in this way, which is a concern, right? And this is characteristic of parasite studies generally. We don't really know where these negative impacts are going to happen. We don't know where parasites are going to be a threat to conservation because they're so unevenly distributed and they haven't been studied enough in order to know what reliably predicts their distributions and therefore what might reliably predict their negative impacts. So if we can't really predict where these parasites are or how common they are and what effects they might have, this raises the question, what happens if they get spread around to sites that they haven't been before? So what I'll highlight now is that our own conservation actions might actually increase parasite-induced population declines. So in the case of freshwater mussels, one of the most popular methods of conservation is translocation. So scientists will identify a site where freshwater mussels are doing really poorly. Maybe there was a previous environmental stress, the population has crashed, reproduction has slowed down. And so what they'll do is they'll pick up a whole lot of freshwater mussels from somewhere else and carry them to that threatened site and put all the new freshwater mussels there, and hopefully, well, hey, that's all solved. However, what I argue is that without thinking about parasites, this has the potential to make things a whole lot worse. So this graph is purely theoretical. It's just a very little simple demonstration of how easy it would be to move parasites around. So on the x-axis there along the bottom, you've got the translocation size, so the number of mussel individuals that might get moved to a new site. And on the y-axis, you've got the probability of a pathogen or a parasite being translocated. And you can see that even at very conservative levels, so if a pathogen is just prevalent in 1% of mussels in that original range, you can see that very quickly the probability of moving that pathogen to the new threatened population rapidly increases. And if you've got parasites at a prevalence, 20%, all you need is basically 10 individuals to get moved somewhere else before you're basically guaranteed to be moving a parasite to. So that little red line there just shows the median translocation size for freshwater mussel translocations. So on average, that's the number of mussels which tends to get moved in translocations. So you can see basically, even if a pathogen or a parasite is just present in 5% of mussels, you're basically guaranteed to be moving that to a new site with unknown consequences. So these are some of the explicit risks of conservation action. You might be transporting parasite-infected mussels without knowing it. The recipient populations could be vulnerable or naive. So going back to the Brandon Creek, which I showed you before, when the mites had the negative effect on Glochidia at Brandon Creek only, Brandon Creek also had fewer mites than the Old West River. So maybe this is a recently new arrival of the mites there. So the mussels at that site at Brandon Creek might have been a bit naive to the mites. They couldn't quite cope, didn't have the appropriate immune response in their gills to deal with this. And that's why Brandon Creek mites were so much worse for Glochidia than they were at the other site. So this is true generally. Recipient populations, if they're receiving a new parasite that they haven't seen before, it was only in some populations but not in others, they might be naive to this, and the parasite might have a significantly worse effect in the new site than it even did in the old site. So this could reduce the effectiveness of conservation action, but it also could make it worse potentially. So that's the theory at least. But what is the actual risk of this? So I've shown you that parasites can negatively affect the reproduction and survival of freshwater mussels, but we don't have that much information on how parasites are distributed among these different sites, as I've said. We also don't know how many parasites there are. So this was a little paper of mine that came out last year, exploring how many parasite species there are in freshwater mussels. So first of all, I should say that this is only for Europe and the USA. And this is because we have absolutely no information on other sites around the world. So this is definitely an example of, the Western focused sampling. In terms of freshwater mussels, there are massive freshwater mussel populations and huge diversity in South America, in Africa, in Asia, and in Australia as well. But there's been absolutely no parasite studies, to my knowledge, in South America, and there's been very, very few in Asia, Africa, and Australia. So this, what I'm going to present next, is limited to Europe and the USA only, but with the acknowledgement that there's probably way more parasites than I present here in these other places too. So what this graph basically shows is the current sampling level of parasites in freshwater mussels. So along the x-axis, you've got the number of freshwater mussels that have been sampled. So unionin is just a fancy way of saying freshwater mussel. Then on the y-axis, you've got the number of parasite and pathogen species recorded from those freshwater mussels. So you can see we've sampled about just over 100 freshwater mussel species, and we've got nearly 100 parasite species from those mussels, so quite a high diversity. There are a couple of problems with this. One, there's over 300 unionid species in Europe and the USA, so we've sampled basically a third of all freshwater mussels. Over 2/3 have never ever been sampled for parasites. And added to this is that 88% of the species that have been sampled are considered undersampled. So not only should our x-axis be increased, our y-axis should also probably be a lot taller, because the species that have been sampled, they've only been sampled, one or two individuals or from one site only. There's probably way more parasites to find in those mussels as well. So really, we're significantly underestimating the amount of parasites that there are. And this is an example using freshwater mussels, but this is true generally. I don't think there's any organism on Earth that anybody could argue has been really well sampled for parasites. Might be a few exceptions for really, really endangered species, but that's about it. So just extending this, using that curve, we can then extrapolate based on current sampling trends, what's been sampled and what hasn't and at what level. We can actually predict how many parasites there might be. So I've broken this up into 4 major groups of freshwater mussel parasites, 2 types of trematode and ciliates and mites. Should say as well, these aren't all freshwater mussel parasites, in terms of there are more freshwater mussel parasites than these 4 groups. There are nematodes, there are leeches, there are chironomids, dragonfly larvae have been reported as being parasitic on freshwater mussels. So there's a whole lot more diversity than what I present here as well. But these are just the groups where there's enough sampling to be able to make a reasonable prediction. You can see Aspidigastrian trematodes. We know that there's 5 different species of these trematodes in freshwater mussels. That's what we've observed. And based on sampling trends, we predict there are actually 6. So we're doing pretty well with these sorts of trematodes. That's because Aspidigastrian trematodes are highly host generalist. So they're not that host specific. They occur over lots of different mussel species. And so given the sampling we've done, the chances of us missing 1 or 2 are quite low. In contrast, you can see Digenean trematodes and ciliates seem to be a lot more host specific. So we've only described, I estimate, about 20% of that diversity. 80% is still undescribed, and mites we've described at best 50%. So, I said we've seen 96 different parasite species just of these 4 groups in freshwater mussels. I estimate there's close to 300 and probably far more than that. So what else might these parasites be doing? I've shown you the effect of 1 parasite trematode species and 1 mite species. There was 289 species, most of which we aren't aware of at all, completely undescribed. The potential threat to conservation and understanding of knowledge on what they're doing to populations, and we just have no idea. But given this massive diversity, is there a chance that parasites are at risk themselves? So I've just shown you that 67% of mussel parasites are undescribed based on the threat risks of their host, because freshwater mussels are also highly threatened. 21% of those parasites are at immediate risk of extinction themselves, because obviously if their host goes extinct, the parasites will also go extinct, which obviously is quite a lot. That's equivalent to about 50 or 60 species that I've just shown you going extinct as well, most of which will have been undescribed. So this could potentially be a conservation concern in itself. Obviously, if we're trying to protect biodiversity, it seems like parasites might be a threat too. And so the question then becomes, from the other side of the coin, what other implications might this have of losing these parasites? So parasites can actually play really important positive roles in the environment too. One of the key reasons for this is that parasites are species specific, and because of this, they can actually maintain diversity. So I've seen that in the same river, different mussel species have quite different parasites. So it's actually very striking. You can go into a river, there's multiple mussel species co-occurring. You pick up 2 mussels of different species, dissect them, and they just have completely different parasite communities. Absolutely wild to me that these 2 mussels living within a couple of square centimeters can have such different parasites. And the implication of this is that this reduces interspecific competition. So just an example of how this works, using our trematodes again. So I've shown you that mussels infected with trematodes can't produce babies, they're castrated, there's going to be no Glaucidia joining the next generation. So if there's no babies, or there's fewer mussel babies rooting into the next generation, we're going to see fewer adults of that particular species. But trematodes need those adults. They're one of the hosts in their life cycle. So if there's far fewer hosts, it's harder for trematodes to complete their own life cycle. And if there's fewer trematodes, then we're going to see more mussels that are able to reproduce in the next generation. So because each individual species, host species, has its own specific parasites, they sort of regulate and bounce against each other. So if one species, one mussel species, declines quite rapidly, then it's going to get less parasites because its own parasites find it difficult to find it. Then it can bounce back and go in these repeatable cycles with other mussel species. So in this way, Trematodes can actually maintain diversity, which is thought to be a general benefit of parasites. Another thing they could potentially do, another way that parasites could be beneficial, is actually enhance ecosystem services. So this is what I was telling you about before, how freshwater mussels are so important for filtering the environment. So this is just a little experiment I did looking at how Trematodes affected this. So on this graph, you can see I've relabeled the axes from the paper. Those aren't the original axes that I said in the paper. But basically on the x-axis, you've got the amount of stuff I fed the mussels. So I basically put lots of algae into these little buckets at different levels. And then I saw how much they ate. So on the y-axis, that's the amount of stuff that the mussels were able to filter out of the water column. And you can see that generally we've got this trend, which makes sense. The more I gave the mussels, the more they filtered out. This is exactly what we want to see. This is them, cleaning their environment, basically. However, what we also see are these two lines there. So the yellow line is uninfected mussels, and the grey line is trematode-infected mussels. And you can see that consistently, for a given level of algae in the water, the trematode-infected mussels actually filter more than the uninfected mussels. So they're doing a better job of filtering the environment. It's sort of weird, but the way that we interpret it was oxygen competition. So these trematodes living in the gonad, they also need oxygen, just like the mussel does. And because mussel gills fulfill this dual function, both of getting oxygen for the mussel and of filtration, if mussels are in competition for oxygen with trematodes, mussels need to take up more oxygen, so they need to filter more water through to get that oxygen through their gills. That also filters more particles out of the water. So on these small scales, trematodes could actually enhance the ecosystem services provided by freshwater mussels. There's a whole range of other ecosystem services. They can encourage food web connectance and stability. So food webs with lots of parasites tend to be more stable than food webs without parasites. They can also concentrate and remove pollutants. So these could be a boon for conservation. Another good example is biological control. One of the key reasons why some invasive species do so well, why they become so invasive in the first place, is that they experience enemy release. So when a species invades somewhere else, it's generally invading in small numbers. Often it's the most fit individuals that are invading, or it's specially selected individuals that are moved by humans to go somewhere else, the best individuals. And this means that they leave their enemies behind. So all their specialist parasites, pathogens, they get left in the home range and they arrive in their invaded range, basically parasite-free. And this is why they do so well. And so there's the potential to actually use parasites as a benefit for conservation by introducing their enemies again to these invasive populations to manage them. So a couple of examples are lungworms and cane toads. That's been somewhat effective at managing cane toad populations in Australia, and obviously myxomatosis in rabbits. That's a disease of rabbits, which was absolutely instrumental in initially wiping out a large population of invasive rabbits in Australia and in New Zealand as well. I'm on the fence about biological control. It's often promoted as a benefit of parasites, or a good way to use them in conservation. It's highly debated, again, because we don't know enough about parasites. So in my opinion, we don't know the host range of parasites. We don't know the risks often of if we move a parasite somewhere else. Could it then affect native species in that new range? Could it be even worse on invasive species than it is? Could it be worse on native species than it is on the invasive species? We just don't know the answers to these questions. So really, from what I've told you, I've got these sort of contradictory sides to this story. The parasites represent two different conservation coins, both good and bad, or are they one side of the same coin? And it's just the way that we look at it. So on the one hand, they could be valuable for ecosystem services. They maintain diversity. they're severely undescribed themselves and threatened themselves, and indeed it's been promoted, something I agree with, that they need a conservation plan of their own. Then on the other hand, they appear to reduce population level viability, or indeed species level viability of often threatened populations, and could actively damage conservation actions trying to help those threatened species. One aspect which might help sort of reconcile this debate or show that it's a matter of perspective is something that I've kept from you so far, and that is that freshwater mussels are themselves parasitic. So these Glochidia, returning to them, you can't really see in that photo, but they have little hooks on each side of their shells, because these Glochidia are parasitic on fish. So these will attach themselves to the gills or the fins of fish, and basically suck the blood of that fish and develop on the fish for a period of time before then dropping off and becoming adult mussels in the environment. The question is, how do these freshwater mussels get their Glochidia onto these fish? So it looks here like this freshwater mussel has captured this fish. So somehow it's managed to lure the fish in and then grab the fish and maybe transfer its Glochidia that way. That photo, what that photo shows is actually all mussel. So that fish that's there, that's just a big extension of the mantle cavity. So that acts as a massive lure. So that looks like a little fish that other larger fish might like to eat. So they'll come and try and eat that fish, but actually that's just all mussel. And when they attempt to eat that, it will burst and basically explode Glochidia into the face of the fish. And that's how the Glochidia can attach themselves to the fish and develop on there. Those are just a couple of other examples of mantle lures that the mussels use to lure in these predatory fish and help transfer their Glochidia. So that's mostly in North America that do that. But this is a European example. They have a different strategy to transfer their Glochidia to fish. So this is Uniocrassus. It's a threatened mussel species in Europe. And what it does, as you're seeing in this picture, is it will crawl up on the bank to the edge of the water. It will poke its exhalant siphon out of the water, and then it will squirt these jets back into the middle of the river. And in these jets, you can see those little white dots there, hopefully in that photo. Those are its Glochidia. So that gives you an example of how small these Glochidia are. But it will shoot its babies out of the water, back into the middle of the river, and then the pattering of the water in the middle of the river, that attracts fish and think, oh, maybe an insect's fallen in, I can go and eat that. They'll go and investigate those patterns and basically swim through then the cloud of Glochidia that's been shot there by the mussel. So there's a couple of images of those mussel Glochidia, those baby mussels attached to the gills and the fins of fish. So there's a whole body of work on how mussels negatively affect fish, how they're parasites and maybe need to be managed, harm vulnerable fish populations. So really what I'm trying to get across with this point, one, hopefully is to show you some interesting natural history. So I think that life history strategy is absolutely wild. But two is that parasitism is sort of a matter of perspective, and that freshwater mussels play important roles, but they're also parasites. Just like parasites might be perceived as negative, but they can also play incredibly important roles. So I would say that parasites aren't a threat to biodiversity or conservation. Parasites are biodiversity. They're an important part of the biodiversity of the world, just like any other species. And what you might notice is that mainly the negative impacts that I talked to you about, that's due to the interaction with human influence. So the negative impacts when parasites become really dangerous is because that's when we move populations beyond historic boundaries. That's when parasites interact with human-induced stressors, things that, parasites that mussels or other organisms might be able to deal with on their own. Parasites then only become bad when you've got warming or anoxia or other stressors. And this is affirmed by a recent analysis of primate parasites. And what they showed is that there's been this massive loss of specialist parasites, these host-specific ones that generally tend to maintain diversity and aren't too bad. But what has increased is generalist parasites, which will spread around a lot more, that are a bit more able to deal with environmental conditions such as warming, that can then have large negative effects across a wide range of different hosts. So the key thing that I think here is that we don't know enough about parasites. They're just like any other species in terms of potentially good and potentially bad, depending on context. For example, the possum is threatened and protected in Australia. It's hugely threatened and valuable species there. But the possum is a major pest in New Zealand and kills all our native birds. So this context is key. And what's important to understand for parasites is that we just lack that context. That's all. That's the only thing that's different between parasites and the rest of biodiversity generally. We just don't know enough about them, and this can seem to be very threatening for conservation when we don't know how best to manage them. So the diversity, as I've shown you for freshwater mussels, and is true generally, is still largely unknown. There's a famous quote by Aldo Leopold with regards to conservation, which says, the first intelligent precaution of intelligent tinkering is that you keep all the cogs on wheels. So basically, before anything else happens, you need to make sure you're at least keeping what there is before messing with stuff. With parasites, we don't know what those cogs on wheels are. That's all. So one frontier is that we need to describe more parasites. We also need to understand how they've changed through time. Just like understanding general biodiversity trends, we need to understand how parasites have changed too. And historical collections might be a valuable and important part of doing this. This is becoming a new frontier in parasitology. There's lots of natural history collections. For example, fish parasites are being majorly studied for this at the moment. There's lots of preserved fish in museums. We can go and dissect those fish, dissect modern day relatives, and see what sort of parasites there are and how they've changed. I want to leave you with this perspective, is that a single animal is an ecosystem. So from my own PhD, I found a maximum 14 different parasite species inside one mussel. That's not one mussel population, that's 14 different parasite species inside a single mussel individual. And this is only macroparasites, so multicellular organisms, Trematodes, mites, nematodes, things like that. This doesn't include bacteria and viruses, other microparasites, which we know are probably very, very important. We just have no information about. And this perspective is key for conservation, that when you're thinking about moving animals, when you're thinking about protecting animals, thinking about their threats, a single organism is actually an ecosystem of all the other biodiversity that lives inside it. And hopefully this perspective can help us better integrate parasites into more effective conservation and recognize them as a valuable part of biodiversity rather than a threat to biodiversity. So that's it. I'd just like to thank my supervisor for my PhD, David Aldridge, and my two advisors, Bill Sutherland and Ed Turner, and my other co-authors on the papers I talked about today, Sam Reynolds, Isabel Ollard, Sebastian Dunn, and Christine Ellis, and of course the Wolf Fisher Trust for funding my research. And thank you to you all again for listening. And I'm very happy to take questions now. Thank you.
Great. Thank you so much, Josh. That was fascinating. I love the twist towards the end. And it's just really, it's great to hear you kind of questioning our assumptions, perhaps, about parasites. So we're going to go to questions now. One of the great things about these events and about our audiences is the fantastic questions that you ask. So please do put your questions in the question and answer box. I can see there are a few already there. So I'm going to go straight to some of those questions to make sure we get plenty of chance for them. So Helen asked a question quite early on. I think this is about the mussels, asking whether they are anodonta. And can you tell us a bit about what that might mean?
Yeah, so anodonta is a genus of freshwater mussels. So I had two major study species. So one was Anodonta anatina, and the other was Unio pictorum. So the two mussels that you saw me holding up at the start, looking very gleeful, they were both Anodonta anatina. Anodonta are particularly interesting in terms of parasites. They have quite a few trematodes, whereas Unio pictorum have no trematodes. So they were the two species that I was thinking of when I talked about, going into a river, picking up two mussels side by side, and they have completely different parasite fauna. it's just amazing that, you know, you pick up this anodonta and it's just stacked with different trematodes, and you pick up a Uniopictorum right next door and it's got no trematodes at all. And it just makes you wonder, you know, how does this happen? It's just fascinating to think about, I think. But yeah, so anodonta is one of the key species that I was studying.
Fantastic. And it opens up a whole bunch of other interesting questions to work on, I guess, doesn't it?
Definitely 3 or 4 PhDs worth of stuff in there that I wasn't able to explore.
Yeah, we'll just have to clone you. Great. So from Ruth, the next question is, what method did you use for identifying the diegenic trematodes?
That was about 3 months worth. So there's almost no keys for trematodes in the earlier stages of their life cycle. So this is a sort of a big gap for parasites generally, I think, is that parasites with multi-stage life cycles like trematodes, there are quite sophisticated keys that exist, but they're only for adults. So if you're dissecting a fish where the adults live, that's fine. But especially for diegenian trematodes, and specifically what I was looking at is a subtype called bucephalid trematodes. So they're the ones which look like, the Dickon balls with the 2 little flagella coming off each side. At that stage, those Sicariae, they look exactly the same. There's about 10 or 12 different species which all have that type of Sicariae, and they're almost impossible to tell without genotyping. So I was able to do it. Fortunately, I had a contact in Finland with probably the world expert on freshwater mussel trematodes. So I was able to send him some of my photos, and also by looking at previous host records, it seemed like in my species, the key trematode of that type was Ripicotyle campanula, and that was the only one that's ever been observed in Anodonta anatina. So basically, by process of elimination, that's what I ended up doing. But yeah, the first couple of months of my PhD, I was just dissecting mussels, taking photos under the microscope, and going, well, what on earth am I looking at here?
Wow, so really, really kind of time-consuming and kind of detailed work then.
Yeah, and it just shows you how little is known about these different stages too.
Yeah. So the next question, there's absolutely loads popping up, so I'll try and get to as many of these as possible. From Ray, there's a question about can ex situ populations of endangered mussel species be established, and does the complex life cycle create problems for reintroduction?
Yes.
A couple of questions there.
Yeah, so they definitely can, and it's only the last couple of years that people are working out how to do this. So that is one of the key things about reintroductions too. So I talk about parasites and host specificity. So freshwater mussels are very host specific in terms of what fish they will and won't develop on. And so a lot of work in the last 10 years or so has been done on basically these massive trials, getting Glaucidia and seeing what fish they do and don't develop on. Obviously, so it's a really good question. It's also hard. One, that's just difficult to try and manipulate Glaucidia. Two, these are often, as you said, endangered populations. And so, you can't do large-scale manipulations. If you're going into a river, sometimes, you'll only find 2 or 3 individuals. And if one is gravid, then that might be the entire Glaucidia for the next generation of mussels in that thing. So it's very difficult to justify then taking that out, doing experiments on it. Well, if you just remove the one mussel that was gravid in that river, it's incredibly difficult. So yes, you can establish ex situ populations if you know what fish they will develop on. And yes, you can do reintroductions, but it is incredibly difficult to do so because of all these difficulties. Again, it comes back to, I keep banging on about the same thing. It's lack of knowledge. And when you get down to this level of endangeredness, difficult to justify exploiting individuals to get more knowledge when they might be the last couple of individuals of a population or of a species. So it's this vicious cycle that we get trapped in when we don't think about parasitism early enough.
Great. And sorry, my dog made a brief appearance there.
That's all right. He likes parasites too, obviously.
Really enthusiastic about them. Great, thank you. So the next question is about, you kind of touched on it already about kind of using DNA. And the question is specifically, can we assess the biodiversity of mussel parasites in the environment using eDNA? And is it possible to find parasites in historical collections as well?
Yeah, so that the second part first. Yes, absolutely. And that's something that a lot of people are working on now. So that's the way forward. In terms of eDNA, yes, like you technically could. I'm sure if you went out into the river, extracted water, did analysis on it, you would get genes and DNA back that was parasite DNA. The problem again is lack of knowledge. So my favorite example of this is when people first started doing eDNA, they went to the Arctic and they extracted things from the Arctic, and they found Asian elephants in the Arctic, DNA from Asian elephants. And they thought, well, what's going on? Oh, this is so weird. Were they suddenly were they far more distributed than we thought? And the answer is no. It's that obviously it was mammoth DNA, but they didn't, in the databases they were searching against, they obviously didn't have mammoth DNA in the database. So the closest relative was the Asian elephant. And so that's what popped up. And so this is the problem with parasites, is that they aren't in these databases. So yes, you could get DNA, but basically there's nothing to search against. So before eDNA is going to be effective, we need to do more natural history, basically, go very targeted, build up these databases to extract parasites, do morphological identification on them, because genetic identification is still so new for lots of these parasites. Work out what species they are, build up a database for each species, and then eDNA will be an effective method. But right now, at least in my study system, eDNA wouldn't be effective for freshwater mussels because there'd be nothing to search against.
Yeah, so you need this kind of research first before that can happen.
Yeah.
Slightly different kind of question now, a really good one actually. From Yasin, I'm sorry if I've mispronounced your name. People eat mussels. How do these kind of parasites affect that? Are there any dangers or anything that we might need to think about?
For humans, no. So people do generally eat marine mussels. At least in the US and Europe, people don't eat freshwater mussels. And I wouldn't advise eating freshwater mussels because of their filtration capacities. So basically all the crap that's coming down a river, that ends up concentrated in mussels. So the marine mussels that you eat are generally also very well cleaned. They come from big mussel farms. They're not wild caught necessarily, and so they go through an extensive filtration procedure before you eat them. So in terms of diseases, potentially it's not so dangerous, but there will be definitely in these mussels, if you ate them, and in marine mussels there will be trematodes and mites and things like that. But even if you eat them raw, they won't be able to develop in humans. So from that perspective, they're not dangerous. But as long as you don't mind the thought of potentially having a few trematodes along with your mussel, then it's fine.
So it's more the other things that might be in the water that you might need to worry about.
Yeah.
Great. So the next question is about globalization and whether that kind of causes generalist parasites to be spread around, and do they cause more problems globally, the generalist parasites, than when specific parasites are spread around the world?
That's a great question. The answer simply is yes. Yeah, if a parasite is a generalist, if it can infect more hosts, if that's going to be moved around, then basically no matter where it moves, it's far more likely to be able to infect a new host, potentially have negative consequences. In general, I would say, again, we don't have much knowledge on the degree to which generalists are moved around more than specialists, but absolutely, from the perspective of if a generalist move versus specialist, that's going to be far more dangerous, which is why it's so important to identify host ranges, work out how many different host species these parasites do exist in and what the threats might be if their hosts are moved.
And it comes back to that need to kind of to understand more about what these species are and where they are and all sorts.
Yeah.
Great. Okay, that question was from Pamela, by the way, sorry. From Neville, what measures can be taken to keep distinct populations separate and avoid infection, if that's something that we would want to do?
Ooh, great question. I think I'm sounding increasingly boring, but it's just know what parasites are in different populations. So that just comes down to a knowledge thing. I think one thing you can do potentially, you could do some DNA analysis of different populations, because phylogeny, so the genetic relatedness of different species and different individuals within a species, tends to predict parasite distribution and diversity quite well. So we could see, the genetic relatedness of different populations, and that might be able to predict whether there's going to be a risk necessarily of moving different parasites around or not. That'd be quite a loose approximation. So I'd say the best thing is just, get your hands dirty, see if you can find out how well distributed these parasites are among different populations, what they have and what they don't.
Mm-hmm. Great. This next one is a really, really interesting one. Is there any prospect of looking at historical parasite fauna through documentary records as well as museum collections? Would people in the past have been able to kind of to tell if these parasites were present?
Ooh, that's a great question.
We found another PhD project for someone to work on.
Yeah, I haven't really thought about that. I would think probably yes. I'm sure there are probably some proxies that would be recorded in writing of being able to predict parasitism. And this comes back to, again, different types of knowledge. So obviously in science, we privilege journal articles and sort of the Western method of doing science. But I think there is so much to be learned from looking at the recordings of different cultures, the way they interact with their environment. So that's a great question in terms of linking that in. But I can't think specifically of what you could do, but I'm sure there are things you could do to link that in. Great,
thanks, John, for that question. From Jeffrey, is there any evidence that mussel innate immunity relates to parasite specificity?
That's a brilliant question. Again, so basically several of the questions that I've been asked are in the future directions section at the end of my thesis. So yes, I'm sure there is. There's been almost nothing done on immunity in freshwater mussels in the context of parasitism. So I probably can't answer that question more than that. I would definitely estimate that they're related. I'd love to see how, but I don't think anybody knows the answer to that question at the moment.
Sorry, there's clearly people knocking on the door. Sorry about that. So next question from Timothy is, if we decide that mussel parasites are a worthy target for conservation, what might this conservation look like? What sort of actions would it involve?
Oh, that's a great question. I think because parasites are so closely linked to their hosts and sort of rely on their hosts by definition, it would look like preserving a healthy ecosystem as a whole. So in the case of trematodes, key number one is identifying all the key hosts in their life cycle. So what mussels can they and can't they use? What fish can they and can't they use? And then ensuring that those different host populations have healthy conservation status. And so those parasites are going to be able to be maintained as well. I'd say that's the key thing, just sort of making sure their host populations are doing okay. And the first step for that is just, again, understanding what sort of hosts these parasites use, how they're distributed, and how important these different host species are.
Yeah, and it comes back, I guess, to what you were kind of saying in your talk about human interactions sometimes being the thing that kind of causes the problem, actually, and trying to keep that to a minimum. This might have to be our last question because I'm aware we're coming up to 1:30. is there any, it's from Janet, is there any possibility that a generalist parasite could curtail the spread of invasive mussels, so such as the zebra or quagga mussels?
Yeah, that's a really good question. Potentially, yes. I'm just trying to think. This was sort of towards the start of my PhD, where I thought about the interaction between invasive and native mussels and their parasites. Definitely. So I think there's actually evidence from zebra mussels that in some populations they have built up their parasites to an extent that they're starting to regulate the zebra mussel populations. But also the opposite could be true as well. So there's one example, Synodonta woodiana is an invasive mussel from East Asia, which has come over to Europe, and that's competent for the parasites that are also found in Synodonta woodiana. And basically what it seems like it's doing is it's then amplifying those parasites. So it can take up that generalist, but it's not harming that invasive population too much. But because it's competent for that generalist, it's then amplifying that in the ecosystem and feeding back more to the native mussels. So yes, so it could definitely go both ways. There's a lot of theory on how invaders could both suffer from generalist parasites, but also potentially amplify generalist parasites. And both of these could be possibilities. So that's another great question. Oh, I think you're muted, Anna.
Oh, sorry, I was trying to stop the dog noises. That might have to be our last question. Thank you so much to everyone for your fantastic questions, and a huge thank you to you, Josh, for your fascinating talk.
Thank you so much for having me.